Welcome everyone to today's session on emerging topics in biomolecular magnetic resonance. As you know, sponsored by Euromar and ICMRBS. I'll draw your attention to the slide. We proceed under the policy of the Gordon Research Conference that there should be no private audio or video recording of the lectures. With the speaker's permissions, we record the lectures and make them available on the ICMRBS YouTube channel, and that is listed, the channel is listed below. All of this information is available on the website for the seminar series, emergingmr.org, as you see here on the slide. We'll have introductions to today's speakers, and then we ask that you post your questions using the Q&A. Do not use the chat because we won't monitor that. So post your questions in the Q&A. At the end of each talk, the introducer will read the questions to the speaker who will respond. At the end of both sessions, we'll close the official seminar, but leave the Zoom link open for more informal discussions as long as people want to talk. Now, before turning things over to Julie Foreman-Kay to introduce our first speaker, I would like to, on behalf of the organizing committee of the ICMRBS meeting, make two announcements. First of all, as you know, we had originally postponed the 2020 meeting until summer of 2021. It turns out it will be impossible to hold a meeting in the summer of 2021, and the tentative decision has been made to just shift everything then by 1 year. So what would have been the 2020 meeting will happen in 2022. What would have been the 2022 meeting will happen in 2024, etc. You can do the math. There will be announcements posted on the ICMRBS website. I think by tomorrow, speakers have been notified, and we certainly hope by summer of 2022, life will be more or less back to normal. The second announcement is we've received reports of phishing attacks in which individuals are contacting speakers or other individuals listed on the ICMRBS website and posing as representatives of the organization or the hotel, etc., asking for PayPal information, credit card information, etc. So please ignore all phishing attacks. If you're contacted by anyone asking for financial information, hang up, and then you can contact us directly to find out if that was for real. But please don't be duped. So with those two announcements, I'll hand things over to Julie Foreman-Kay.
Thanks, Art. So it's a real pleasure to introduce our first speaker, Tanja Mittag, who is at St. Jude Children's Research Hospital in Memphis, Tennessee, in the Department of Structural Biology. Tanja did both undergraduate and PhD studies at Johann Wolfgang Goethe University in Frankfurt, Germany, and her postdoctoral training was with me at the Hospital for Sick Children in Toronto, Canada, making it really particularly nice for me to introduce her today. So she was really a colleague more than a trainee, and I have learned a lot from her over the years. And recently, I even had the opportunity to publish with her again on SIC1, the main topic of her postdoctoral work with me in a collaborative paper with Claudio Gradinaro focused on SIC1 ensembles with NMR, SACs, and single molecule FRET data. So Tanja's had a number of honors and awards, and it was recently announced that she will receive the 2021 Michael and Kate Barani Award of the Biophysical Society, recognizing her foundational contributions to understanding the biophysics and functional relevance of biomolecular condensates, particularly the role of disordered protein regions. And Tanja's also made important contributions to structural and functional studies of disordered proteins in general. So she's going to speak today on understanding the molecular interactions underlying liquid-liquid phase separation of low complexity domains. Tanja.
Thank you so much, Julie, for this very kind introduction. And it's, I was had fun when I heard this morning that you would introduce me. So that's a great pleasure. Thank you so much. And thank you for the organizers for giving me the chance to present a bit of our work today. My lab is interested in understanding the role and function of so-called membraneless organelles or biomolecular condensates in cells. We often reconstitute very simple versions of them in vitro to understand the underlying interactions, to get insights into their function, and also how they're related to disease. And today I want to talk about how liquid-liquid phase separation, or the ability to undergo liquid-liquid phase separation, is encoded in low complexity domains. It's become clear that liquid-liquid phase separation is really a foundational principle that underlies the compartmentalization of cells and results in the formation of all of these membraneless organelles or biomolecular condensates, and it's important for many functions in cells. And it's a well understood physical process in which macromolecules demix into two phases: a dense phase that is rich in the macromolecules and a coexisting dilute phase. And this can be influenced by parameters like temperature, concentration, pH, salinity, binding partners, and so on. And this liquid-liquid phase separation is mediated by multivalent interactions. This can be encoded in proteins with many domains, modular binding domains, and that would then interact with binding partners with multiple motifs that interact with these domains. And this is relatively well understood because we know very well from seminal work from Mike Rosen's lab, for example, that the higher the valence, the lower is the so-called saturation concentration, so the concentration above which one first gets a dense phase. But phase separation can also be mediated by intrinsically disordered proteins, and how this is encoded in the sequences of IDPs is much less well understood. But dysregulation of phase separation is also linked to disease processes such as cancer and neurodegenerative diseases, and therefore it's really quite important to understand how phase separation is encoded in the sequence of these IDRs. We work on one of the systems in the lab that we work on is the human RNA binding protein hnRNPA1. It's really an archetypical RNA binding protein and is a component of stress granules. It has two folded RNA binding domains and an intrinsically disordered so-called low complexity domain because of its very low sequence complexity. It's mainly composed of small polar residues, glycine, serines, and then interspersed with a few aromatic residues and charged residues. And we showed a while ago that the low complexity domain alone or is sufficient for mediating phase separation in vitro. We can look at it by NMR spectroscopy and see that it's indeed intrinsically disordered. It has a very narrow chemical shift dispersion and a sharp line width. This is work by Ivan Perrin in my lab. You can also look at secondary structural propensity and see that basically there is no secondary structural propensity. This is a really disordered protein. It makes these nice droplets that can fuse well, and we can make dense phase samples, make a large enough drop that it fills the volume, the visible volume of an NMR tube. And when we look at spectra of the dilute and the dense phase, we see that they are very similar, that even in the dense phase, this protein is intrinsically disordered. There are some small chemical shift differences, but there doesn't seem to be large scale ordering. And I'll just point to work by Nick Fauzi, Louis Kay, and Julie Foreman-Kay, who've done really nice work on dense phases of proteins by NMR spectroscopy. And there's more work also from our lab to come, but we're not quite ready with that yet. I'll just say that, of course, as you could see also from the line widths, there is a strong difference in the R2 relaxation rates. They are much higher in the dense phases, which is due to the higher viscosity and intermolecular interactions in these dense phases. And so doing experiments in dense phases is hard. There are these high sample concentrations and it's hard to make the samples, broader lines, And we also think there are lots of interactions due to crowding or due to the high concentrations, and we may not see only the interactions that we are interested in, the interactions that drive the formation of the dense phases. So can we find these important interactions when we do experiments on light phases, on dilute phases? And the idea is the following. The same interactions that drive liquid-liquid phase separation via intermolecular contacts should in systems where a single protein can mediate phase separation, these same interactions should also be operating in the dilute phase intramolecularly. And when we do our two relaxation experiment, transverse relaxation experiments on HNR and PA1, we see here interesting distribution of relaxation rates for a disordered protein where the relaxation rates are only determined by segmental motions of the residues in the chain. We would expect a plateau in the middle and lower relaxation rates at the termini. When we fit this to such a model, our data to such a model, and we see a pretty poor fit, and with lots of outliers, and also the persistence length is has non-physical values. So this doesn't work. But inspired by a model by Harald Stalbe's lab, we can say that there are clusters of enhanced relaxation rates on top of this simple Gaussian model. And then we see a much better fit. And we can see that these clusters center on aromatic residues in the sequence. And these observations are consistent with the idea that there's clustering of aromatic side chains in the sequence. So we wanted to see whether we can directly see these contacts via NOEs. As you can imagine, these low complexity sequences have very overlapped resonances of their side chains, but we can take advantage of the fact that phenylalanine and tyrosine residues have different chemical shifts. So we were not able to individually assign, site-specifically assign phenylalanine and tyrosine side chains, but we can say if we sit on the phenylalanine plane here, then we see NOEs to tyrosine side chains. And if we sit on the tyrosine plane, then we see NOEs to phenylalanines. So there is these direct contacts. We look at the sequence of the protein again, we actually see that we have a tyrosine and a phenylalanine close and directly adjacent in the sequence. So to make sure that we didn't just see NOEs between these two residues, we made a sequence that has perfectly spaced aromatic residues. They could still undergo liquid-liquid phase separation just fine. And we again see these NOEs, which gives us confidence that we actually see clustering of aromatic residues in 3D space. I also want to note that the intensity of the NOEs goes up when the temperature goes down, when we go in the direction of liquid-liquid phase separation. This is a sequence that undergoes upper critical solution temperature phase transition. So as you lower the temperature, there is a stronger driving force for phase separation. So when we lower the temperature, we have higher NOE intensity in agreement with there being more persistent interactions or more frequent interactions between aromatic residues. So this was a collaboration with Rohit Papu's lab and Alex Holhouse, who was a postdoc in Rohit's lab at the time. He now has his own lab at Washington University in St. Louis. He did all atom Monte Carlo simulations with the absence implicit solvation model. And they were in really good agreement with our small angle X-ray scattering data on the protein. And when he looked at which residues made a lot of contacts with other residues in the sequence, the aromatic residues stood out. Nearly each of these peaks is at the position of aromatic residues. And a network analysis of how aromatic residues interact with each other shows you that they're not just one-to-one interactions, but they can pretty much all interact with each other, just that the termini, the aromatic residues at the termini interact with some fewer residues than in the middle, as would be expected. So it really looks like that the aromatic residues might be these adhesive elements in the sequence. to test this and whether they could change their dimensions, whether if we change the number of aromatic residues, that would change the dimensions of the sequence. We either added 1/3 aromatic residues or removed 1/3 or 2/3 of aromatic residues from the sequence. And when we then analyze the global dimensions with small angle X-ray scattering, I'm showing you here a normalized Kratky plot. It really nicely shows that, first of all, the protein is disordered, but then also here the where this plateau, the slope of this plateau tells us something about the dimensions of the protein, and you clearly see that reducing the number of aromatic residues increases the size. And you see this here also in the quantitation. And that really tells us that aromatic residues give rise to cohesive interactions that determine the chain dimensions. Now, I want to also tell you about some newer work, so I'll make this here very brief and just say that in collaboration with Rohit and Alex, or they did a parameterization of a stickers and spacers model. Rohit has introduced this framework of thinking of stickers and spacers, both for folded domain motif type systems and also IDR systems, where a sticker is an adhesive element that can form physical crosslinks, non-covalent physical crosslinks between elements of a chain. And spacers connect these stickers and influence their ability to interact with each other. And they have previously published analytical models where the valence of the stickers determines or is an estimate of the saturation concentration. But this can also be come as a coarse-grained model where not only the valence of the stickers, but now, of course, also their patterning and in the sequence can be taken into account. And they used our experimental data on the dimensions of these different variants of HNR and PA1 to parameterize sticker-sticker interactions, sticker-spacer interactions, and spacer-spacer interactions. And knowing their strength, they were then able to do simulations where you put a lot of chains into a box and do Monte Carlo simulations. And you can see either condensation into a network of chains or the lack of condensation. And this is here for these 3 different sequences that we were working with. And one can quantitatively analyze this and get phase diagrams and compare this with experimental phase diagrams. So we've done here. Ivan Perrin measured these phase diagrams. Here's the experimental data. And when we compare this with the parameterized stickers and spacers model, get very good agreement. And this also explained to us why this arrow minus variant never showed phase separation in vitro, because the critical point is actually below freezing. So the results showed us that the valence of the aromatic stickers determines phase separation, but also their patterning. And there are some interesting effects on material properties that you can look up if you'd like to. I'd rather like to move on to some new work that we've been doing that we put on bioRxiv a few weeks ago, where we're saying, well, we've been treating all of these other residues other than aromatic residues as spacers, but are they really all equal? Probably not, right? And there's probably some more complexity to it. And we get hints of that when we look at the conservation of spacer properties in homologs of HNR and PA1, but also across the fat family of proteins. We see that they have a preference for a small positively net charge per residue. And there's also a decisive preference for arginines over lysines in these sequences. That tells us that this is probably important. And of course, there has been previous work from other groups, for example, from Julie and Husan Chan, but also Rohit and others, and Tanja Mittag. And it's not quite clear yet, I think, whether we can conceptually understand what charged residues do, under which conditions do they make favorable electrostatic interactions? Do arginines always make favorable cation-pi interactions with aromatic residues? And perhaps we can conceptually understand this. And we could also take the other stance and say our null hypothesis is really that the big solvation volume of charged residues, so here the glutamate solvation volume is much bigger than, for example, of leucine, that this big solvation volume really leads in enhanced solubility of proteins, and we'll treat them as high excluded volume spacers. So to address this question, Anne Bremer, a postdoc in my lab, made a lot of variants, charge variants of HNR and PA1. In all of these variants, she kept the number of aromatic stickers and their position in the sequence constant, and just varied the number and position of charged residues. And we'll look here at phase diagrams again, where we see dense phase concentrations and coexisting light phase concentrations. And when she removed more and more, or replaced more and more lysine residues, more and more arginine residues, sorry, with lysine residues, she saw reduced driving force for phase separation. And when all arginine residues were removed and replaced with lysines, we didn't see any phase separation up to a concentration of 2.3 millimolar. This is in agreement with what others have seen, and it really points to our original idea that lysines function as excluded volume spacers. We look then at arginine variants. So here she either added 7 arginine residues, which resulted in weakened phase separation. When she removed 6 arginines from the original 10 arginines of the sequence, we actually got better phase separation, which would suggest that arginines are just excluded volume spacers and weaken phase separation. But when she removed all natural 10 arginines in the sequence, phase separation was also weakened. And this now suggests that, or shows us that arginines can contribute to phase separation in certain sequence contexts, can act as stickers. We looked at aspartate residues to get to the role of negatively charged residues. She added 8 or 12 aspartate residues. Phase separation was slightly or strongly weakened, but when she removed the 4 natural aspartate residues, phase separation was also weakened, which shows that aspartates also have a sequence, have an effect that is strongly dependent on the sequence context. So the big question was, can we understand this conceptually? And can we, if we look at a sequence that we don't know, based on the number of oppositely charged residues, can we understand how strong its driving force for phase separation is going to be? And the idea was that, well, we know that folded proteins have their lowest solubility at the pH that matches their PI, because they have a net charge of 0 there. So if we now look at our set of sequences that don't change in terms of the aromatic stickers, but change in their net charge per residue, do we also see the lowest solubility, meaning the highest driving force for phase separation, of a net charge per residue of 0? So when Meena and Rohit plotted our saturation concentrations here at a single temperature as a function of net charge per residue, well, we see perhaps a little bit of a V-shaped plot if we squint, but there are certainly lots of outliers here, the arginine variants and the lysine variants. So we seem to be missing some information for the stickers and spacers model, and not only aromatic residues are stickers and not all of the spacers are the same, if we believe that this V-shaped plot should hold. So they made use of an analytical stickers and spacers model and said, instead of just aromatic residues as stickers, we will assume that arginines are also stickers that make constructive interactions with aromatic residues in the sequence. And they renormalize the saturation concentrations, and now the arginine variants fall very nicely onto this V-shaped plot. But the lysine residues are, of course, still strong outliers. And we tried several different models, and this model is the one that worked best, where now the lysine residues antagonize aromatic and aromatic arginine interactions in a 3-body manner. And now they collapse quite nicely on the V-shaped plot. There are still some outliers here, and we think this has something to do with patterning of the arginines and lysines in the sequence. But of course, you know, we have used these variants to parameterize the model. So we made a bunch of other variants. Anna and Wade did this in my lab. These variants were not used to parameterize these analytical models, and they fall quite nicely onto this V-shaped plot. So what we learned from this is that the net charge per residue can strongly modulate the driving force for phase separation, you know, up to 2 or even 3 orders of magnitude, and does so by mean field electrostatic effects. We also learned that arginines can be auxiliary stickers in the sequence, and we wanted to see whether we would see NOEs. So we have here a 13C resolved aromatic aliphatic nosy spectrum. We're sitting on the tyrosine and the phenylalanine plane here, of the side chain plane, and we see some weak NOEs, and these go to the arginine side chains in the sequence. So we see arginine aromatic NOEs. We see a few other NOEs, but not many. So for example, interestingly here to this asparagine residue. Wade in my lab wanted to look at this variant plus 7K, plus 12D, so quite highly charged compared to the natural sequence, but with a pretty optimal net charge per residue for phase separation, and did relaxation experiments on this variant. And interestingly, if we compare this to the wild type, we see that these clusters with enhanced relaxation rates are pretty much in the same place as for the wild type, showing that aromatic residues really remain the most important stickers, and that constructive electrostatic interactions are really not showing up as enhanced relaxation rates and restriction of motion. So we learned that aromatic residues are the main contributors to phase separation. Arginines work as auxiliary stickers, but may not work constructively for phase separation if the net charge per residue leads to them being more of a excluded volume spacer that enhances solubility. Lysines antagonize arginine aromatic and aromatic interactions, and negatively charged residues function as excluded spacers. I didn't show you data on glutamates, but they, in our hands, have very similar effects as the aspartates. We also learned that the net charge per residue strongly modulates the driving force for phase separation via mean field electrostatic effects, and we can understand this from the sequence and predict it. So just to say this in the end, we use this mean field net charge per residue term, or Rohit and Meena did this and incorporated it into the coarse grain stickers and spacers model. Also parameterize the arginine aromatic interactions. I'll just show you here one movie of this variant 7R12D, and we see that experimental data and stickers and spacers simulation data are in very good agreement for these number of variants that were not used in the parameterization of the model. So now we can quantitatively predict phase behavior for a large set of these variants, and also for fat family low complexity domains. And I think importantly, we understand better how phase separation is encoded in intrinsically disordered proteins, and that they are not so different from proteins with domain motif systems. And with that, I would like to thank people in the lab who did the work. I tried to call them out during the talk, but I think I didn't call out Eric, who did a lot of the work on the original stickers and spacers model. This was, as I said, a collaboration with Rohit's lab, without whom we wouldn't understand some of the things that we understand now. So this is a very nice collaboration with a good friend and good colleague. Meena and Alex were important in these collaborations. And then there are other people in my lab. And I acknowledge funding. I'd be happy to answer questions.
Thanks so much, Tanja. Beautiful story. So we have a couple of questions already. Rob Tico asks too, what is your interpretation of chemical shift differences between dilute and dense phases? Yeah, very good question. So, of course, we've plotted them. It isn't so obvious what is going on. We see some chemical shift changes of aromatic residues, which you would then say, well, that points to the aromatic residues being the important stickers there, but there are also alanines that change chemical shift. Perhaps we see that some areas in the sequence are more important than others, but overall, when we change the sequence context and shuffle things around, as long as we keep the sequence well mixed, we actually still get really good phase separation that is not so different from the original wild type sequence. So
You know, I think one thing that is going on, of course, is that there are really high concentrations of these LCD molecules in these dense phases, and that there are a lot of nearest neighbor effects that then play roles and modify these chemical shift values. And which is why we like this approach of looking at the dilute phase and characterizing interactions in the dilute phase.
Thank you. So he asks another question. Is it possible that some of the intermolecular interactions in the dense phase could be qualitatively different from intramolecular interactions in the dilute phase? For example, in register parallel alignment of glutamine-rich segments might favor intermolecular beta sheet-like interactions that would not be possible in individual molecules because simultaneous alignment of multiple glyn residues can occur intermolecularly but not intramolecularly.
That's a very good point. You know, I think he gives a very good example where that may be the case, right? That intramolecularly, the same interactions are not possible as intermolecularly. I think I would say we have the hypothesis that in a protein that remains largely disordered in the dense phase, we should be able to see the important interactions intramolecularly. And I think the fact that we could parameterize this model, that we could assign aromatic interactions from a set of variants in which we altered the number of aromatic residues but didn't touch the other residues in the sequence. And that model was then predictive for other sequences, shows us that we're capturing the important interactions, or perhaps the most important interactions. There are, of course, other contributions. And in our newly, I should say, in our newly parameterized model, there are still, although we're now taking into account these different types of spacers in terms of charged residues, there is still a spacer-spacer interaction. So I'm sure there are other things that contribute, but we think we can capture the interactions that drive phase separation most strongly. And, you know, one other point I want to make is in terms of capturing intramolecular versus intermolecular interactions, what I didn't go into, but is in the bioRxiv paper, is that for these charged variants, we don't see a one-to-one correspondence between global dimensions and driving force for phase separation. That breaks down when we change the net charge for residues. But we understand in which way it comes through these mean field electrostatic interactions, and we can correct for that. And that allows us to use the information from single chain dimensions again to parameterize a model that is predictive.
Okay, thank you. So we have another question from the polymer physics perspective. These results are thrilling on the one hand and rather analogous to various types of transient gels, which are also characterized by aggregating non-polar, often aromatic moieties forming cross-links. I struggle with making a more direct link. What are your highest concentrations in the phase separated state in terms of volume fraction or weight concentration? And do you know the overlap concentration and where you are with regards to this common reference point in polymer solutions? This is from Salwakter K. I mispronounced that, I'm sure.
Yeah, so very interesting questions, and I'm not sure I can unpack all of this now. This may take a more in-depth one-to-one conversation, but I can give you some overall thoughts, of course. So where are we in terms of overall concentrations in the dense phase? That depends on the temperature, of course, right? So some of our NMR experiments in the dense phase are like at 40 degrees Celsius, where we get nice and sharp lines, and the concentration is something like 20-25 millimolar there, which amounts to a concentration of about 300 mg per mil. So very high concentrations. It's of course a very interesting point to ask, is the concentration that high in actual condensates? We are looking at very simplified systems, right? We're interested in the concepts of how phase separation is encoded in these sequences. There is indication that once you add RNA and lots of other proteins, that they're going to become more dilute and are not going to be as dense. The overlap concentration is much lower. We see actually that at temperatures above room temperature, our light phase concentration is above the overlap concentration. So they see each other relatively well already at lower concentrations.
Okay, so there are many, many questions. Maybe I'll go. Does Art, did you have a question you wanted to ask?
Sure. So Tanja will recall that at her PhD thesis defense, I was only allowed to ask one question. So I've been waiting many, many years to ask you a second question.
You know, Art, you keep saying that, and I have no recollection that you were only allowed to ask one question.
But when one sees videos of phase separation, one's struck by the time dependence of the formation, merging, dissolution of the phase separated droplets. What do you think it will take to understand those sorts of kinetics of the system beyond, the kind of equilibrium phase diagrams?
Yeah, this is a difficult question because there are so many states, right? And how does one get information on this? I think single molecule techniques, single molecule fluorescence techniques in the dense phase is probably going to help us understand this. What are the lifetimes of the interactions? This is very important for understanding the material properties of the interaction. So on the other hand, one can also come from the mesoscale perspective and measure material properties of the dense phases. And then based on the valencies and the interaction strength, calculate back the lifetimes of the interactions. We certainly, see effects when we change the sticker strength, that we see differences in how quickly dense phases form, which must have to do with the lifetimes of individual interactions and how fast this whole network of interactions can be reconfigured.
Okay, so there's a question from Fleming Hansen that I would also really like to know if you've ever seen any arginine NOEs, and he prefaced it by saying, "Very nice talk, Tanja." So.
Thank you, Fleming. Nice to hear you, hear your question. No, we have not, but we haven't looked for them. So if we looked for them, perhaps we would see them. I can't really answer.
Okay, so Abhishek Kalarikal, another mispronunciation I'm sure, asked what's the reason behind the broad NMR peaks for the dense phase?
So there is a high viscosity in the dense phase. We're at 25 millimolar protein, 300 mg per mole protein. And I think this is really nice data in a paper by Julie Formiquet and Louis K. So this is their work that I'll reference here that shows that it's not only the high concentration, but also the intermolecular contacts or the contacts in general that are in the dense phase that increase these R2 relaxation rates. They had a nice matched experiments. Julie, jump in and correct me if I'm wrong, but they had a nice matched experiments of a protein that doesn't undergo phase separation, but at the same concentration as the dense phase, and then they didn't see quite the same enhancement of R2 relaxation rates.
Okay, so I'll let Louis correct that, but maybe we'll move on. Another question from John Bushweller: have you mixed C13 and 15 labeled LCD with unlabeled LCD and looked at intra and intermolecular NOEs via half filter to see if you can detect intermolecular NOEs?
Yeah. I'm not sure whether the question is with respect to the dense or the dilute phase. So actually we have done it in neither of them. But there are some examples in the literature. I'm thinking of data from Nick Fauzi's lab where they have seen this and they see, and I think also data from Julie and Louis where they see lots of intermolecular interactions in the dense phase. And we wanted to take a slightly different stance and say, well, there's so much protein in the dense phase and basically, everything is close to everything. And perhaps that's why there are so many NOEs. And can we rather identify the interactions that drive phase separation if we look in the dilute phase? So no, we have not done these experiments.
Okay, thanks a lot, Tanja. And it's my big pleasure now to introduce Adam Lange, who's the second speaker here. So he studied physics at the University of Göttingen, then did his PhD and postdoc studies in my department together with Mark Baldus, who was a solid state group leader in the department at that time from 2002 to 2006. He was so successful that he won the Ernst Award of the German Chemical Society and the Otto Hahn Medal of the Max Planck Society for his PhD work. Then he continued as a postdoc with Beat Meyer, mainly at ETH for 2 years, was very much educated there in fibrils, came back and was then a group leader here at my place from 2008 till 2014. And when he won an ERC starting grant in 2013, he immediately moved away to join Berlin, the FMP, where Hartmut Oschkenat was the first, and up until then only together with Phil Zelenko and Leif Schröder. So he joined there and is since 2014 now department head at the Leibniz Institute for Molecular Pharmacology, FMP, in Berlin. He won in 2016 the ICMRBS Founder Medal. And I think he's known for his PhD work on toxin-induced toxin interactions with KCSA. Also some technical experiments like the NHHC and the CHHC experiment. Then he got involved in aggregate investigations, alpha-synuclein and tau. That, of course, he became perfect in that then in the postdoc with Beat, and during the time here then as a group leader, I think the work sticking out most was the structure determination of the type 3 secretion system needle, which was kind of right at the time before cryo-EM took over and then was solving all these structures, and of course also on membrane proteins like VDAC. So long introduction finished. Adam, please use the floor.
Yeah, hello everyone, and thanks a lot, Christian, for the very kind introduction. Thanks to all the organizers for inviting me. It's a great pleasure and also an honor to speak here today. And as Christian has mentioned, we are in my lab interested on the one hand in supramolecular assemblies. So we have solved a couple of atomic resolution structures. It was the type 3 secretion needle. We worked on the type 1 pilus and later on also on cytoskeletal filament up to filin. And a current project that I would like to discuss today is related to the bacteriophage tail tube, where we actually combined NMR with cryo-EM. On the other hand, we have always been interested in membrane proteins. And today I would actually like to talk about two projects. One deals with selective and non-selective cation channels. And I would like to tell you a little bit about what NMR can contribute to understand the differences in selectivity on the basis not only of structure, but also on the difference in dynamics. And finally, I would like to introduce another cool system for NMR spectroscopists to play with, which are intramembrane proteases. So I would like to start now with the bacteriophage tail tube. And here you can see in this artistic representation how bacteriophages attack a bacterium. And they then inject their DNA into the bacterium where they replicate, and at some point the bacterium explodes and releases new bacteriophages. And you may be aware that since antibiotics are facing the problem of resistance, bacteriophages are actually a hot candidate as an alternative. And in fact, in Eastern Europe, for example, they have been used for many decades as a means to treat bacterial infections. And we produced actually this tail of the bacteriophage in vitro by producing the protein called GP17.1 that forms the tail tube. Here you can see a negative stain EM of purified GP17.1 after 3 weeks of polymerization. So all you have to do is produce the protein, put it in the right buffer, and let it sit on the bench for a couple of weeks. Then you have these beautiful tubes formed. And from previous studies of our collaborator with whom we do this project, Sophie Zin-Justine in France, we know that there's hexameric rings which are stacked on top of each other with a distance of 40 angstrom, and they also rotated relative to each other by about 20 degrees. So we have these global parameters, but the local details are missing. And in the beginning, we did this project only with NMR. Later on, we combined this with cryo-EM, which I will show you in a minute. First, I would like to show you now a spectrum of GP17.1 in the polymerized form. So this was done using our favorite probe, which is a 1.9 millimeter 4-channel probe on the 900 megahertz magnet. And you can see that you have actually very good resolution. So you see all these peaks. However, this is a fairly large protein. GP17.1 has almost 180 residues, and they are not disordered in the assembled structure, so they all contribute to the spectrum. And so we need to figure out which peak is what, of course. And we have in the beginning done this with 3D experiments, and for this we use this approach here that has been developed by many people in the field, and we have summarized that here in this user-friendly protocol. But however, unfortunately, just using 3Ds alone, we were not successful in getting unambiguous and complete resonance assignments. So we had to extend this by going to a 4th dimension. And so actually already in 2014, in the time back in Göttingen, we proposed some 4D proton-detected experiments that allow to get unambiguous assignments of large proteins. And in this case, we designed experiments in which you can get 4D spectra connecting H, N, C alpha, and CO within one residue, but also between two residues. And so you have always two connections to link the residues. And Max Zinke, who is the PhD student in my lab, who actually does all of this bacteriophage project, he then extended this approach also to include C beta. And using this, then in the end, he was successful in obtaining complete and reliable assignments of all of the residues in the spectrum. Now, one very important aspect of determining structures of supramolecular assemblies is, of course, to understand how these different subunits pack relative to each other. So you need to determine intermolecular interfaces. And if you know about carbon-detected experiments, there's different means to do this. So there's different heterogeneous labeling schemes. For example, you can label some subunits with N15, some others with C13, and then you mix them and you look for C13 and N15 contacts. But with proton-detected experiments, there's actually not so many possibilities. And so we have designed some schemes that make use of methods. So here you see one example where we have labeled all the isoleucine C delta ones, both with C13 and protons in an otherwise deuterated and C12 labeled background. And as you can see here in the sequence, isoleucines are found at regular distances through the sequence. And if you look at the spectrum, the carbon-proton correlation spectrum of such a labeled sample, you see that it looks actually quite neat. So, and what is also very nice is that in this case, even each isoleucine C delta 1 has a unique proton frequency. So for example, if you look for the frequency here of isoleucine 18, you see that this is a unique proton frequency. And then you can go for 3Ds, where you take in the 3rd dimension this unique frequency in protons, and then you connect it to an NH plane. And for example, here in the case of isoleucine 18 H delta 1, then you can get a lot of contacts, and most of them are actually found close. So you see 17, 18, 19, 20, and this allows you also to assign this frequency to isoleucine 18. Max has actually also done some more complicated things, so he has really connected with experiments the H delta 1 frequency to the NH frequency, but you can also get this information but just by analyzing which peaks you see here. And then you see some additional peaks. But of course, in such a sample, you don't know which one is intramolecular, which is intermolecular. And for this, you have to go now to the heterogeneous labeling. So here we mixed H delta 1 labeled or method labeled GP17.1, which is not N15 labeled, with other subunits which are exclusively N15 labeled but not method labeled. And now you see nicely that there are 3 peaks which are intermolecular, and you know that now we have an interface between isoleucine 18 of one subunit to these residues in another subunit. And we haven't done this only for isoleucine, but we have in fact done this for all the possible methods that you can imagine. So we have this for leucine and valine together, but also for alanine, threonine, and methionine. And we then in the end combined the information. We got all these methyl-based restraints, but we also got traditional 4D-HNHN restraints and combined this with a medium resolution cryo-EM map. So you may ask, so why don't you do this just with cryo-EM? The reason is that the bacterial phages that we were interested in, they have a flexible tail tube, and that kind of limits the resolution in the cryo-EM to around 4 angstrom in the best parts, but in the flexible parts, it's much worse. So just using cryo-EM alone, you would never get the structure. Also, one has to be true and say, you have to be honest and say just with NMR, you would also not get it. But if you combine EM and NMR, you get the answer. And I'm showing here already the final result. Here you see the structure. So you have kind of rigid rings. And you can actually get the structure of the rigid rings also by NMR alone. We have done that in the beginning. But then you see that you have connections between the rings via relatively flexible linkers. These are very difficult to look at by EM, but what is important is that you have, for example, some contacts between the rings that are not easy to identify just by NMR alone. So for example, this contact that I just talked about on the previous slide between isoleucine 18 of one subunit, that's somewhere here, with the other residues, which are more in the C-terminal extension. We knew from our NMR experiments that this were intermolecular restraints, but of course we can't really tell. We can only tell it's an intermolecular restraint, but we cannot really say this is a restraint from subunit I to subunit I plus 7. It could also be to I plus 8. And this is where then cryo-EM comes into play. But if we combine it, we get a unique solution and an atomic resolution structure. And I should mention that the EM was done by Gunnar Schröder in Jülich, and that we had to actually develop further some protocols originally developed by Michael Nielges, also with Michael Habeck together, that basically allow you in a Bayesian approach to combine the information from different sources. Okay, now I would like to switch topics a bit and come to the membrane proteins. So I will start with the cation channel NAK. You may be familiar with a more typical channel like KCSA that conducts potassium. But as the name suggests, the cation channel NAK allows you to conduct sodium and potassium. And I would like to first summarize a bit what is known about KCSA. It's a highly potassium selective channel. It has a conserved selectivity filter sequence TBGYGD. And here you can see the structure that you are probably all familiar with. Now the channel that we study is called NAK. It's homologous to cyclic nucleotide gated channels that are medically quite important. And importantly, it's a non-selective cation channel. So it allows for the conduction of potassium, but also sodium and even divalent ions. And the surprising thing is that the selectivity filter is not so different from KCSA. So there's just 2 residues which are different. So the tyrosine here in KCSA becomes an aspartic acid, and the aspartic acid becomes an asparagine. Otherwise, the structure is fairly similar. Now, if you look at the NMR spectrum, You see some surprising effects if you compare to results obtained on KCSA. And one thing is that there's a gating hinge, so the channel allows to change its structure between an open and a closed conformation, and below the gating hinge, all residues are invisible in our spectrum. And so that indicates that these regions are dynamic if you look at the protein in a lipid context. And also the upper part of the selectivity filter is not visible in this NCA spectrum, further indicating that this is also either disordered or dynamic. And then in contrast, this lower part of the selectivity filter is visible in the spectrum. However, we do see peak splittings, and that is also something that had never been observed for KCSA, for example. So you see that there's two conformations at least for the lower part of the selectivity filter. Now, here I would like to briefly say something about methods development. So this NCA spectrum here, this is something which is a traditional carbon detected spectrum. You also have to get the assignments, and traditionally this has been done, for example, with NCOCX experiments. They have a very poor sensitivity, and some years ago we introduced this band selective homonuclear COCACP to improve the sensitivity of this experiment. And I would just like to mention that here, because we have last year published a small paper, but it's a very long paper, which I find very nice because it basically provides a very detailed tutorial how to do these 3D experiments to get assignments. And whenever I have a new student in the lab, master student or PhD student, they start with this protocol paper and they sit for a couple of weeks on the spectrometer and learn to do their experiments themselves. So I can just recommend you maybe to have a look at this paper. Now, what do we do with this NAK channel? The first thing that we tried was, of course, since this is a channel that conducts sodium and potassium, to prepare samples with different ions. So we started to look at the channel without any ions. Then we actually get two conformations. And one conformation is more populated, and that is this conformation we call the ion-free conformation. Now, if we add sodium, then the ion-free conformation is still the mainly populated conformation, but it furthermore splits into multiple subconformations. So you see further peak splittings, and that indicates that sodium actually causes a break of the symmetry of this tetrameric channel and causes some kind of heterogeneity. In contrast, if you add either potassium or rubidium, which is similar to potassium, you get the other conformation to be more populated. So this is the conformation that we call potassium-rubidium favored conformation. And if we want to understand now what's going on, we can combine actually NMR with MD simulation. But before, I would like to present a control experiment that we did. So we can mutate this NAK channel, which is non-selective, into a potassium selective channel simply by introducing two mutants that bring the selectivity filter back to the sequence that KCSA has, to this canonical potassium selectivity filter structure. And the spectrum that we obtain then is shown here. Here we get actually something that looks very similar to KCSA. So I was actually surprised to see that even the pattern, for example, of the glycines here looks remarkably similar to the spectrum that you get for KCSA. So the structure must be highly similar. We see, in contrast to NAK, where we see the whole selectivity filter, and we also see that there's no peak splitting in the lower part. So that really tells us that there's not only a difference in structure between the selective and the non-selective channel, but it's really the dynamics which are different. So only in the non-selective channel, you have these different conformations, you have the absence of the upper part. So the non-selective channel is much more flexible and can really adapt to these different ions, whereas the selective structure is not able to adapt, for example, to sodium, and therefore it is not permeable to sodium. But we have then teamed up with Han-Sun here at FMP, and she ran some MD simulations. So these are actually computational electrophysiology simulations. We have two double bilayers with 2 membrane proteins inserted, and then because of periodic boundary conditions, you have one compartment in the middle. The upper and lower parts are connected, and then you can put more ions here than here, and therefore you get an outward flow, and in the other direction you get an inward flow of ions, and you can really simulate what's going on with the ions in the selectivity filter. And here you see what's going on for potassium. In this case, it's inward flow, and here you see what's going on with sodium. And on the right-hand side, you see this a little bit better. You see that the actual way how the ions flow through the filter is different between potassium and sodium. So for sodium, there's also a side entry that is used, and that cannot be found with potassium. And also, what is maybe even more interesting is that potassium goes through the filter when all subunits are exactly in the same conformation. So it goes in a symmetric way through the filter, whereas sodium only permeates when there's a break of the symmetry. We have summarized that here in this scheme. So as I mentioned before, the selectivity filter of NAK adopts 2 conformations in the absence of ions, and sodium prefers one and potassium the other. And potassium permeates through the entire narrow pore and single file, whereas sodium permeates through a unique side entry ion conduction pathway. And I should mention that very recently we have been able to see this side entry also crystallographically. So this is not an artifact from the MD simulation, but this is something that you can really capture also in crystal structures. And in my opinion, the key to the non-selectivity is the structural plasticity of the selectivity filter, and the selection then of these conformations by the different ions. Now, one side aspect of this project actually deals with the question of how potassium ions are conducted. And for this purpose, we went to the selective variant of NAK, which is called NAK2K. And so you are probably familiar with this picture here that resulted from Roderick McKinnon's work, where there's always a water molecule between 2 potassium ions. And then if a new water comes from below, it pushes everything up, and you come to this situation, then another ion comes, and you have a cycle where water and ions are co-transported. So this was the traditional view, and it was challenged in 2014 by work of Berthe Groth in Göttingen, who proposed that there's actually no ions, no water between the different ions. And this was also supported by crystallographic analysis by George Sheldrick in Göttingen. And yeah, we were thinking, how can we contribute as NMR spectroscopists to this debate? And it is actually very difficult to look at potassium with NMR. It's not impossible, but it's difficult. But what is quite easy for us is to look at the water molecules. And so we thought, okay, let's see whether we can locate water in the selectivity filter. And for this, we first started with HD exchange experiments. And we first started with a sample where there was potassium in the buffer, and so there was potassium ions in the selectivity filter. And what we found was that there's actually in the lower part of the selectivity filter a region which is not accessible to HD exchange, and that indicates that there's no water. And as a control experiment, we washed the channel. So we washed the channel in a buffer which did not contain any potassium. So we washed out the potassium, and then later we put the potassium back, and then we had actually HD exchange here. So that tells us that it's in principle possible to get HD exchange, but as long as potassium ions are around, there's no HD exchange, and this indicates already that there's no water molecules. And a bit more compelling evidence comes from these water to protein magnetization transfer experiments by spin diffusion, where we look at magnetization transfer from water molecules to amide protons. And as you can see here in this summarizing slide, summarizing panel, you see that there's a lot of transfer going on in the vicinity of 2 structural water molecules. But in the vicinity of the sites here, S1, S2, S3, S4, there's no transfer basically. And all the transfer is explained by these structural water molecules. And what I find interesting as an NMR spectroscopist is that you actually see also differences in the behavior of the buildup. So there's some positions, for example, this glycine 47 here, which is close to the bulk water, where you have this just increase. And that the reason for this is that the amide at glycine 47 is in contact with the water molecule, which itself is in contact with the bulk water. And so the relaxation is dominated by the bulk water. And then here you have some other cases where you see that the signal decays, for example, the ones that are close to the structural water molecules. And the reason for this is that now the relaxation is actually dominated by the behavior of these of the static protein. Okay, so in our view, there's no evidence for water in the selectivity filter, and that fits nicely to simulation results. These were actually done in collaboration with Bert and Wojciech in Göttingen. And here you can see again a simulation which shows you how potassium goes through the NAK2K selectivity filter. You also see nicely here in the simulation the two structural water molecules, but there's no water in between the ions, and that fits very well to what we see from NMR. The last thing I would like to discuss today deals with intramembrane proteases. So these are really interesting proteins. So the basic scheme, the basic idea is summarized here. You have this enzyme, this intramembrane protease, which is a membrane protein, and then you have a substrate, which is also a membrane protein, and usually you have a soluble domain. This is anchored via this linker to the membrane via this helix here. This helix in the membrane, the enzyme comes, cuts the helix, and then the soluble domain is released. It swims away and starts a signaling cascade. And there's different classes of these intramembrane proteases. They can be found in all branches of life. They are very important. One famous example is actually the gamma secretase that cuts the protein APP. And you know that the fragments then are these A-beta peptides that form aggregates in Alzheimer's disease. So it's also these intramembrane proteases are interesting pharmacological targets. So it's very interesting to see how they work. And the interesting aspect here is, of course, that you have an enzymatic reaction going on, and the active center is hydrophilic, but the reaction happens in a hydrophobic environment. So water molecules have to come to the active center, and the substrate itself is hydrophobic. So how does that actually work? And I think in this quest NMR can really contribute, and we have started with a protein called GLUB-G from E. coli. This is a serine protease. It has a histidine and a serine in the catalytic center. And you can see here the NMR spectra. This is a carbon detected spectrum. Here you see a proton detected spectrum. The resolution is actually quite nice for a membrane protein. So you get 0.1 ppm for protons. For carbons, it's less than 1 ppm, around 0.7 ppm. So this is something that you can really work with. And we have also obtained most of the assignments and started to analyze the dynamics. And now this is something now where we can start to understand how this protein works. So in our hands, it looks like this protein, when it sits in the membrane, is mostly in a closed conformation. So this is all in the absence of the protein, in the absence of the substrate. And sometimes it opens, and then the substrate can enter the active site, and then it closes again. And now we can start to play. This is something that we are doing currently. So we can, for example, lock this gate with a disulfide bridge and see what's going on, or we can actually loosen the connection between these helices. So then we can actually see in activity assays that the enzyme becomes even more active, and we can relate now the differences in activity to differences in dynamics parameters that we probe by NMR. So that's a quite fruitful protein for NMR spectroscopists. And with this, I would like to finish my presentation today. I would like to acknowledge all the co-workers. So I talked a lot about the bacteriophage tail tube. That was work from Maximilian Zinke here. Then I started to talk about NAK. This was work from Chao Wei Shi here, and also from Kitty Hendricks. Later, Karl Oester joined, who did also mostly the study with the detection of the water molecules in the selectivity filter. And he is also involved in the work on GlybG, which was also established in my lab by Chao, and where now different people in the lab work on, for example, Claudia and Pascal. And I should also mention that Sasha is producing a lot of good proteins for us, and that a lot of the methodological work also has very strong contributions from this guy, which is Benjamin Shebekov. And with this, I would like to finish. I think I mentioned all the collaborations within the talk, and I'm happy to take your questions.
Yeah, thank you very much. Start with the questions. Now I have to see which ones are for you. So far, I mean, so we are still waiting for questions for Adam. So let me just start with two, which I which I collected from the talk. One is in the first project you had this this tail which was going from one monomer to the other monomer. It was indicated in red, and I mean, I guess the tail cannot be seen in cryo-EM, but the tail probably will also not be visible to which segment then it binds. So I was wondering, how could you locate that tail?
Yeah, well, actually, fortunately, one could see this. The resolution is not very good, but this is actually a good example because you can still see with cryo-EM to which subunit it goes.
Okay. Okay.
But you don't really have, I mean, you don't have the resolution to really get atomic details in any way because it's quite flexible. But by the combination, so we have atomic resolution restraints from NMR, but we don't know to which subunit it goes. But this could really be solved by EM.
Okay. Then, okay, now questions come in. So Rams asked, great talk, Adam. Can you provide some details on the sample lipid protein ratio? On the sample lipid protein ratio, model membrane, are they liposomes or whatever, and NMR sensitivity for the 4D experiment?
Yeah, so yeah, thanks for the question. So unfortunately, the 4D experiments were mostly done not on membrane proteins, but on these supramolecular assemblies where we have no lipids and where the rotor is packed with a protein of interest, so we get good sensitivity. And we have actually on the GlybG protein, we have also we have done some 4Ds, but not the ones that I showed. They were all on supramolecular assemblies, but usually we take around 50 lipids per membrane protein.
Okay.
So of course, I mean, usually we start with more and then we go down and see at which point the spectrum changes, indicating a structural change. But usually we have something like 50 to 1.
Okay, Rams will ask further questions if he's satisfied. If he's not satisfied, then a question by Flemming Hansen. Very interesting. Have you tried to use 15N ammonium as a probe of potassium in the solid state, potentially to measure contacts to the protein or exchange?
Yeah, so we have, yes, the answer is yes. So we have done it and it looks interesting, yes. It's something in the making. But since this is all will be all published here on YouTube, so I didn't show it today, but it works.
Okay.
Actually, I mean, it has been done before by Roland Rieck in solution NMR. There's a JAX paper where he looked at ammonium in KCSA, but with solid state NMR it actually works quite well. So you can see the different sites in the spectrum.
Then Umit Akbay asked, nice talk, Adam. Always impressed with the nice resolution you get with your proteins. Is the
Yeah, so I guess the question
Wait a
is,
second. What, 9 millimeter? 1.9 millimeter. The favorite probe. Is your favorite probe the 1.9 millimeter and measurements with fractional back exchange samples?
yeah, so maybe I forgot to mention this, but in the spectrum that I showed for the GP17.1 protein, this was actually a fully back exchanged protein, so fully back exchanged in water. And the resolution is quite good. I mean, if you go to 1.3 or even 0.7, of course, you get nicer resolution, but of course you lose sensitivity. And also since these 0.7, for example, we also have a 0.7 probe here in Berlin. Theoretically, now it's been with Bruker for several months because it has to be fixed. And also the rotors are horribly expensive, so it's not my favorite rotor. So I think I prefer somehow the 1.9. We have done a lot of stuff with the 1.9. It's very robust and you can do a lot of good stuff with it.
Rams is continuing. Are you using fluid lipid membrane to measure enzyme action via NMR?
No, I mean, what I have meant is that we are just, I mean, there's some activity assays. So what you can do is, I mean, there's a lot of different possibilities, but you can nicely probe the activity of the rhomboid protease by just putting together the substrate and the rhomboid protease. And then you see, for example, in the gel I mean, since the activity or the reaction of this enzyme is to cut another protein into two halves. And then you can see this actually on a gel, for example. And you can then compare and there's also fluorescent assays, which are a bit more quantitative, and then you can relate the activity of the enzyme with the dynamics that you detect by NMR. So we want to understand really like how the dynamics, there's, for example, this lateral gate, this opening. And the question is, I mean, if we now make the connection between the two helices that form the gate more loose by, for example, mutating hydrophobic connections to less hydrophobic connections so that it opens more often or that it stays open longer, and then we can really probe now with our activity assays how that relates then to the activity. And so we can come up with some really kind of picture of how this protein works. And that, I think, brings us one step forward in comparison to static pictures that you get from crystallography. So, like, of course, there's tons of structures of this protein. So we're not interested in the structure of GlypG, really, but we really want to see how it functions, how it works.
Okay, I go to the last question before we start the informal session, and then people, please just raise your hand and you will be admitted to the panel in order to discuss with the speaker. So the last one is by Elias Akuri. Great talk, Adam, and very interesting topics. Did you try different percentages of D2O in the HD exchange experiments, and how would D2O contribution fit to the MD simulations?
Yeah, it's a good suggestion, but we haven't really gone for this. We haven't really done it yet.